In the last video, we have discussed about virus cycle and some of the types of penetration. Uh, in this, we have discussed three types of penetration, or we would say like two type of penetration. One is direct fusion penetration, and another one is receptor mediated endocytosis. And within receptor mediated endocytosis, there are two types. One is for the virus, uh, that is virus that has envelope and that penetrate via receptor mediated endocytosis, and virus that does not have envelope and that penetrates via receptor mediated endocytosis. So in this video we will discuss about how the virus binds with the cell virus membrane binds with the cell membrane in case of enveloped virus, that is virus virus that has enveloped proteins. Proteins like this one, this. This is the spike protein. This is the envelope. This is the membrane. I can write it. Spike protein, envelope, membrane, and this is nucleocapsid. In case of virus that does not have envelope, in that case receptor in that case the receptor mediated endocytosis work in a way that a part of nucleocapsid will lysate the plasma membrane plasma membrane sheet that helps in that covers the virus when the endocytosis occurs. Like when the endocytosis starts, it starts like this, and it's come in within the cell. This is the coating of plasma membrane. In case of virus that does not have envelope, this nucleocapsid directly come in contact with the coating of plasma membrane, and the component of nucleocapsid has the ability to lysate this coating of plasma membrane, and so that now the protein, not the protein, now the genetic material of the virus comes within the cytosol, and now it can be replicate and transcribed by the cell machinery of the cells. However, today our focus is on the envelope proteins. Envelope containing virus. So, in order to understand it in an easy manner, this is considered as a cell membrane or plasma membrane. Or this is virus membrane. It has some fusion proteins in its this fusion peptides, you can say more precisely fusion peptides that are highly unstable, and when they come in contact with, they come close to the cell membrane. And also when the pH is acidic or very low, then they change, there is a conformation change occurs in low or acidic pH. There is a conformation change occur. The conformation change occur and it anchors. The first step is called prefusion step. And when in low acidic condition, due to conformation change, it anchors with the cell membrane, and this is called anchoring of fusion peptide. Fusion peptide. When it anchors to the fusion peptide, now the post fusion state of the post fusion state, highly stable post fusion state is different than the pre-fusion state, stable pre-fusion state. So in order to reach to its highly stable post fusion state, it has to go through hemafusion states first. Intermediate state is hemafusion state. So now what this anchor is trying to do is it tries to contract both of this membrane and bring this membrane come in contact with each other, such that one of the bilipid layer of the cell membrane makes a bond with the one of the bilipid layers of virus membrane. This is the hemafusion state, and the final state, that is post fusion state. This is the post-fusion state where anchors almost come in contact with both of this anchor come in contact, both of this opposite end of the anchor come close to each other. That is post-fusion stable state. So now what happens? The same thing happens on the other side. So this resulted in the pore formation on the cell membrane, and from this pore formation, the virus can go inside the cell membrane. So this is how the virus, enveloped protein virus, penetrate inside the cell membrane, that is in cytosol. So this is penetration step for receptor mediated endocytosis for the viruses that have envelope. Thank you.